Welcome to my course Aspects of Biochemical Engineering. Now, as you know that most of the engineering branches that usually develop from the science branches, because science basically tell us that why it is happening and how it is happening, and engineering is the application of science. So, if you look at the chemical engineering, that has been developed from the chemistry, like physics, that we have electronics, electrical engineer, then we have material science, we have development of metallurgy engineering and mechanical engineering. So, engineering is basically that is deals with the application of kind of science. So, I will be discussing on the different aspects of biochemical engineering, and this biochemical engineering mostly deals with the different biochemical processes. The first I try to explain what are the different biochemical processes, and then I want to give you the difference between the chemical engineering and biochemical engineering. Then also I try to tell you that what are the how this biochemical process and chemical process they differ from each other, and finally I want to discuss the details of this course content. So, initially that let me tell you that what do you mean by biochemical process? Biochemical process is the essential part of many food, chemical and pharmaceutical industries. Now, if you look at this is the food, chemical and pharmaceutical industries. Let me give the example that if you look at the food industry, we know that cheese making industry. Now, cheese making industry, what we are basically doing, we produce the cheese for the preservation of milk and protein, which is very good diet, very important diet for our health. So, so this is the process through which you can preserve the food. And also sauerkraut, there is another kind of product, the fermented product, through which we can preserve the vegetables in the natural color, because the vegetables, if we preserve in a longer period of time, then either we go for drying process or use some kind of preservative. If you go for drying process, it undergoes some kind of Brownian reaction, and due to Brownian reaction, color of the vegetable will be changed. That is undesirable. But if you in the sauerkraut, we use some kind of lactic acid bacteria, and lactic acid acts as a preservative, where the green color of the vegetable we can preserve for longer period of time. Then I want to talk about the chemicals. Then if you look at the different biochemical industry that can involve for the producing different chemicals like lactic acid, citric acid, we have ethanol. So, different type of chemicals can be produced through this biochemical processes. Now, and in the pharmaceutical industries, we produce a lot of antibiotics, because in the day-to-day life, when we have some health problem, we go to the doctor, and doctor prescribe some kind of antibiotics for curing the disease. Now, biochemical process make use of how these products are formed by using the microbial cells, animal cells and the plant cells. These are the different living cells. They can be used for producing this kind of products. And not only living cells, some of the biomolecules which produce from the living cells, that may also use for the production of certain chemicals. As for example, I meant that enzymes which is usually secreted from these particular living sources, that can be utilized for producing different type of products. So, I can give a typical example that high fructose corn syrup, where glucose is converted to fructose. Why glucose is converted to fructose? It has two advantages. One is the fructose has more sweeter than glucose, so but for diabetes patient it is mostly recommended. And another thing is that for the preparation of the medicinal tonic, that if you use the glucose as the medicinal tonic, as you know, it comprises of some amino acids and vitamins which are very much heat sensitive. So, naturally we shall have to preserve this at a low temperature in the freeze. So, at low temperature, when you have the sugar syrup or glucose syrup, you will find some kind of crystal formation of the glucose will take place, which is undesirable. Now, if we have the fructose syrup, then fructose even at low temperature, it will not crystallize, it remain in the liquid form. And another thing is that it is more sweeter. So this is the desirable characters that we have. That is how it is used mostly in the confectionery industry as well as pharmaceutical industries. Now, next is the biochemical engineering. What is biochemical engineering? Biochemical engineering may be considered as a multidisciplinary field that implements the engineering principles to design and operate the unit processes required to successfully produce high quality bioproducts. Because that for the operation of the process we required some unit processes. What are the unit processes we have? As for example, bioreactor, because in the bioreactor is kind of unit process. We use some kind of pumps and the sterilization, some that different purification also we have. So different unit processes are involved to get the different pure products. So this biochemical engineering will deal with everything. This is how these different this unit operations that can be handled. So and another is the chemical engineering principles plays a central role. Now this is to be highlighted that if you look at the chemical engineering principles plays the central role to produce the biochemical products on a large scale for marketing in purified form. So basically we are using the chemical engineering principles in this process. Now question comes how you are applying the chemical engineering principles, because we know the I told you that chemical engineering develop from the chemical reactions. And chemical reactions like this, A plus B reacts to give C plus D. Now when it produces the product, we can develop some rate equation. When we develop the rate equation, we have rate constant, order of reaction. From that we can do the reactor analysis. Through the reactor analysis we use this rate equation, and then we find out the volume of the reactor. So this is how we can for suppose we want to produce 4 tons of citric acid by using Aspergillus niger. So that this can be produced. We can find out through this analysis that what will be the size of the reactor for getting the 4 tons of bioproducts, both in the batch process as well as continuous process can be done. So these are this information that we want to give you through this project. So, and at the end of this project you will be developing the expertise for doing this particular analysis. Now next is that I want to give you the differences between the chemical engineering and biochemical engineering. Now chemical engineering, if you look at that, it deals with the synthetic or the chemical processes. As for example that I can acid and alkali, when they react with each other, they form the salt. This is, or if you I consider when burning of coal in presence of oxygen, when coal burns, it produces the carbon dioxide and water. This kind of pure chemical reaction that we have. Or if you look at nitrogen and ammonia, in presence of catalyst, it produces the nitrogen and hydrogen. When it's combined, it produces the ammonia, which is largely used in the fertilizer industry for the production of different fertilizers. Now these are the purely chemical processes that we have. In the biochemical process, they mostly deal with the biological organism and their biochemical pathways. Now biological organism, they are very sensitive. They are very sensitive, and that every organism, they have the metabolic pathways. As for example, if we take glucose, it goes through the metabolic pathways, what you call Embden-Mayer pathway, and through the Embden-Mayer pathway it produces the pyruvic acid. And this pyruvic acid, when passed through the TCA cycle or Krebs cycle, it produces the carbon dioxide, and this is how we get the energy in the form of ATP, ADP or NADH, like this different energy molecules forms in our body. So, so this is the, and if you look at the metabolic pathways, then you will find the different metabolites formed during this. So we can, if we manipulate this pathway, we can easily fish out our desired product from the this metabolic pathways. So that this is how this biological organism can be nurtured for the producing different kind of products. Then it in the chemical process is involved the design and operation of industrial manufacturing plants, because how in the bigger plant that it can be handled, those kind of information that chemical engineering gives. And in the biochemical industry we study the behavior of the living cells, that as for example that I can give you that living system, that behavior of the living system is something different as compared to the non-living system, because living systems should be carefully handled, and this can be handled only if we develop the feelings for the living system. I can give you a typical example that if you look at the relationship between mother and the baby, and mother and the baby is with the mother from the 0 hours. So mother has great feelings for the baby. So anything happen, it happens to the baby, mother always understands what is the problem with the baby. But so same thing, when you handle any kind of living system, we should also develop kind of feelings for the living system, otherwise we cannot handle. We should know under what circumstances it should work in a proper manner. So it's not like non-living systems, they just you add A plus B and give the product C plus D. It is not like this. So you have to handle your organism. Only the difference is that in case of chemical process, suppose we want to produce any kind of product, you have to have the specific raw materials is required for product formation. But in the biochemical process, it's totally different, because from a particular simple compound like glucose, we can produce n number of products. The glucose can be converted to citric acid, glucose can be converted to acetic acid, glucose can be converted to lactic acid, glucose can be converted to ethanol. So n number of products we can produce. Only what you do, we change the organism, the living organism. So living organism that name, as for example, when we produce citric acid, we use the Aspergillus niger to convert the glucose to citric acid. When we use the lactic acid, we use the Lactobacillus delbrueckii, which convert the glucose to lactic acid. Now, when we use suppose acetic acid, we use the Acetobacter aceti, which convert the ethanol to, the first initially the acetic acid ethanol is produced through the Saccharomyces cerevisiae, which convert glucose to ethanol, and then subsequently when you use the Acetobacter aceti, then the ethanol is converted to acetic acid. So what I want to highlight, that basic difference between the biochemical and chemical process is that the chemical process, one raw materials is remaining more or less same, and only by changing the organism we can get the n number of products. In the chemical process, as your product changes, your raw material changes. If your product is complicated, your raw materials will be complicated. I can give a very typical example. Another example is antibiotics formation, like penicillin. If we want to penicillin production, that we use the glucose in the fermentation broth, but at the same time we use some kind of precursor like phenyl acetic acid or phenoxyacetic acid and get the product like this. So, but if you use in a chemical process, first of all, penicillin is very difficult to produce chemically, but if you use some kind of complex chemicals, your raw materials will be also complex. As your raw material will be complex, your cost of the raw materials also will be very high. Now, in the chemical process, another thing that we have, the reactants form products in the presence of chemical catalysts, and in the biochemical engineering, the product formation takes place with the help of biological catalysts. Now, what is the difference between the chemical catalyst and the biological catalyst? Chemical catalyst, we have platinum, palladium, nickel, they can take part in number of reaction, different type of reaction they can take care. But if you look at the biochemical catalyst or biological catalyst, they are very specific. As for example, if you look at glucose isomerase enzyme, they can act only glucose and produce fructose. They cannot convert fructose to glucose. So they are very specific as far as substrate is concerned. Now, if I like protease that only act on the protein molecule for the degradation of proteins, it will not degrade the starchy molecules. So they are very specific as well as substrate is concerned. Now, chemical reaction occurred at high temperature and high pressure, and since it is required high temperature and high pressure, naturally it is energy intensive. As now in case of biochemical processes occurred at ambient temperature and atmospheric pressure, because now this is very important, because so since it is ambient temperature and as atmospheric pressure, we are less energy intensive. The energy point of view, this biochemical process is very attractive. Also substrate point of view, also very attractive. Substrate, we required mostly the glucose, sucrose type of material which is largely available, and not only largely available, that is renewable, that can be, we can produce again and again through the agricultural product formation. So if we look at the biochemical process, it can be divided into three parts. One is feedstock. Feedstock may be anything, it may be gas, it may be liquid, it may be solid. Now bioprocessing, we have biocatalyst, we have bioreactor and recovery of product. So now here we require the cell, that microbial cell, it may be plant cell, it may be the microbial cells, it may be the animal cell, anything we can use, and then we do the product purification. So we can broadly divide it into three parts. One is this is the main part where the reaction take place, that where the biochemical reaction take place, and before the biochemical reaction take place, we shall have to prepare our feedstock, and this whole unit we call it upstream processing. And then after the product formation, then we call the purification of product. So it can be divided into three parts. One is upstream processing, bioprocessing, then downstream processing. Upstream processing, which feed the raw materials to the process, and in the downstream processing, it mainly deal with the purification of products. Now biochemical engineering principles can be applied in three different areas. As for example, fruit processing. Food processing, I already explained how it is used in the food processing. I have given the example of cheese making industry and the sauerkraut. Then agricultural production, that we largely use. As for example, I can give the example of bioinsecticides. That because that usually we use the lot of pesticides in the agricultural to increase the agricultural production. Now pesticides is such a chemicals, if we take in our system, it accumulate in our system which has the carcinogenic property. But bioinsecticides, that it's something different. It is kind of delta protein, and which also has that kills the pest. So if you use the bioinsecticides, then what is happening? Even we take in our body, it is kind of protein that will not harm in our system. So that is how it can be used in the agricultural sector. Now another important product that we have, the GM food, that is the genetically modified food. And as we are living in the sparsely populated country, that a time will come when our land will be insufficient to produce the significant amount of desired amount of product. So naturally we shall have to reduce we shall have to try to use the food in a manner so the small amount of food will be good enough for our survival. So this is possible if we enrich our food. As for example, particularly we know in case of vegetable food, the vegetable protein, the utilization efficiency is very less. Why it is less? Because the essential amino acids in the agricultural protein is less. and that reduce the utilization efficiency. So, if we increase that, if whatever amino acid, essential amino acid is the deficiency is there, if we increase that particular protein, the utilization efficiency of the protein will increase drastically. This is how we can improve biochemical way. Now, this biochemical processes can be used for the production of different chemicals. I have given already the examples of ethanol, citric acid, acetic acid, lactic acid, all these things, different type of butagonic acid, all can be produced through the biological means. The healthcare, I have given the typical example of different antibiotics like penicillin, largely used. You know that due to invention of penicillin, it is becoming easier to cure our infection, particularly all the athletics, all the soldiers, when they involved with kind of interaction, they have some kind of injury, and during the injury they have some pus formation due to the infection of the gram positive bacteria. By using penicillin, it can be cured. So, this has a lot of antibiotics nowadays we use if our health has some kind of problem. We go to the doctor prescribes some kind of antibiotics for the rectification of that particular disease. Now, energy sector, that biochemical process has a greater role to play, because the fossil fuel resources will be exhausted very soon, because this has the limited reserve. So, different renewable energy sources are explored. Bioenergy is one of the area paying a lot of attention as far India is concerned, and as you know that particularly in the gasoline, that government recommended that 10 to 20% the gasoline is to be replaced by the ethanol. Now, if we reduce then our gasoline consumption will be reduced significantly. Then environmental protection, there is another very important area, because we have a very great concern, because particularly we can think about the Paris Convention, where most of the country they agreed to safeguard the environment. Particularly, I want to talk about this carbon dioxide content in the air, and India also signed the agreement that we should control the carbon dioxide in the air, so that if we increase the carbon dioxide content in the air, the temperature will increase, what we call the greenhouse effect, and due to this increase of temperature, there is a possibility of melting of ice in the northern hemisphere. Then that causes the hike in the rise level, and we know some of the western country is almost at the sea level. So, if the sea level increases, those countries will be under this water. This is a great concern throughout the world. So, we have a lot of applications that we have with the biochemical engineering principles in the different areas. Then what are the advantages of the biochemical products? That also I want to highlight, that first important thing is that this is sustainable. This is sustainable means this process we can have for a longer period of time, because the raw materials that is used, that will be available with us, because that will not be exhausted very soon. And another very important thing is that less carbon and water footprint. Less carbon and water footprint is very important, because water consumption, because we know that as the time passes on, that water consumption will significantly decreases, and this carbon and water footprint plays very important role. Create the rural employment opportunity. This is very important. The reason is that this can be used for the smaller scale and bigger scale application. Less emission of pollution to the environment. This is also very important, because pollutant which is coming out, that should be as low as possible. Biodegradability is and recyclability is very important. I can give the example of plastics. Is not biodegradable, but you use the bioplastics, the biopolymer, this can be easily biodegradable, so it can be it will save our environment. And high productivity and raw materials from the local sources, that also very important, because if it is available from the local sources, then we can use it very easily. So, of course, the objective is that we want to use the basic concept of the microbiology. We use the basic concept of the microbiology, biochemistry, chemical and biochemical reaction kinetics. Also we applied, so initially we will start the course with microbiology. We will give the preliminary information on microbiology, then with the biochemistry and chemical and biochemical reaction kinetics. This is the different information we will give, because we will give more stresses on chemical and biochemical reaction kinetics to understand the process much in a depth, and then apply the chemical engineering principle in the bioreactor analysis and design, upstream, downstream processing, and bioprocess optimization and control. So these are the different areas we'll cover in this particular course. Now, scope and relevance of the course is several. We have biochemical engineering mostly deals with the complicated life system. I have already explained that what you mean by complication life system. The life system, we shall have to develop some kind of feelings for the process, otherwise we cannot handle the process very nicely. Then describe the behavior of any life system in terms of mathematical form. I told you that mathematical representation of a process is called mathematical modeling. We try to give the develop some kind of modeling of the living system, and also we want to simulate the validity of the mathematical model comes through the simulation of the process. Now, it is an interdisciplinary subject where all the different disciplines like biologist, physicist, chemist, all these people will, all these experts they will help each other to develop this subject. So this all knowledge is required to understand the process in a better way. Then we have the different transport processes, that is very important, transport processes that is involved, and sophisticated advanced control besides the conventional biology, that also we are going to explain in this sense. So this course will deal everything, all this material in details. Now let me give you the details of the course content. Initially we talk about as I told you, microbiology, biochemistry, classification of the bioproducts, because as far bioproducts is concerned, we have different types. We have low value products, high value products, medium value products. So different classification products we will talk about. Then we shall discuss the stoichiometry of the bioprocess, that is also very important. Thermodynamics of the biochemical reactions, which is very important just to understand what is the nature of the reaction, whether it is spontaneous or it is unfavorable reaction, or also we can find out to what extent the reaction take place. The thermodynamics can help us to understand the process in a better way. We'll discuss about the kinetics of the homogeneous system and the heterogeneous system. Both the system we will discuss in details. Now, in case of heterogeneous system, the homogeneous system usually take place when the reaction take place in one particular phase. Heterogeneous system, that one phase has to diffuse to the other phase, then and only then the reaction take place, and after the reaction is over, the product has to diffuse from that phase to the other phase. So diffusion, the phenomena is to be considered during this particular heterogeneous reaction. So we will be kinetics of the enzyme catalyst reaction. Then we'll discuss about the enzyme catalyzed reaction using the free enzymes and using the immobilized enzyme, both we can discuss. And then kinetics of the substrate utilization, product formation and biomass production. This is very important aspects as the biochemical processes are concerned, where we use the living cells. And then design and analysis of the bioreactors and scale up of the bioreactors, that is a great concern, how from the lab scale we can switch over to the plant scale and a bigger scale, that we will discuss in details. Transport phenomena of the bioprocess, that how the flow system, different flow system, I want to take the momentum transfer, mass transfer and heat transfer, how take place in the system, that I want to discuss in details. The heat and medium sterilization, this I told you that in the biochemical system, The one important that we want to grow our desired organism. To grow the desired organism, we know our environment comprises of different other organisms. So if the sterility we don't maintain, then we cannot allow our organism to grow in that particular reactor. So sterility is very important factor as the biochemical processes is concerned. This I shall discuss in details. Then downstream processing and effluent treatment processes, process control and biochemical process and operation of the industrial fermenter, and finally I shall do the summarization of the whole course. Now the overview of the course is like this. If you look at this, I told you that upstream processing, and it deals with the strain selection, because the kind of microorganism that is available, that is in two different forms. One is the wild strain and there is the industrial strain. Wild strain we cannot use for industrial fermentation process because they are very unstable, and the industrial strain, they have the genetic stability, so they have reproducible characteristics. The medium optimization, there is another, and feedstock collection and sterilization. All these things undergo the upstream processing. Then in the fermentation or biochemical processes that involve the enzymatic process and the microbial process. In the enzymatic process we have free enzyme, immobilized enzyme, and the microbial process we have suspended cell and the immobilized whole cell. And also we use the recombinant cells. Recombinant cells, so suppose we want to produce a special type of proteins, then we go for the recombinant. I can give the example of insulin production, that is usually done with the help of recombinant cell. And in the downstream processing, basically it deals with the cell disruption, solid liquid separation, concentration, purification, formulation, and the effluent treatment. All these different aspects will be discussed in the downstream processing. So this course will give you the total idea that how the biochemical process can be analyzed and desired that and operate. So hopefully that this course will be very much useful to all of you, and different references, book references I have given here. So these books you can consider, particularly Levenspel. This is chemical reaction engineering. I shall discuss about the chemical reaction kinetics and reactor design. All these things will be available here. Then Suller and Kargi, this is bioprocess engineering, this is also very important. Then Bunch and Glark, that is biochemical engineering. Bailey and Ollis, biochemical engineering, fundamental Duran, bioprocess engineering. And Doctor, this is principles of mass transfer and separation processes. This will be used for understanding the transport phenomena. Thank you very much.